Thank you so much. I'm Eric Horne. I'm a senior director on the Med Affairs team and patient advocacy team for Avidity Biosciences. And today I'll talk about, oops, oh, where are my slides? There we go. Perfect. Thank you. Okay, there we go. So I'm going to talk today about our initial results from our phase 1/2 trial of the Fortitude study for AOC 1020 in patients with FSHD, right? And so at Avidity, we're really committed to profoundly improving the lives of people through the delivery of RNA therapeutics, right? My forward-looking statements. So I will be talking about some of our results. These are the results as we know them today, and it's driving some of our decisions, but our decisions may change. So that's the forward-looking statement. So at Avidity, we're really anchoring on developing the AOC platform. This is an antibody oligonucleotide conjugate platform, right? One of the biggest challenges of getting these different RNA therapeutics to muscle cells is getting them past the liver, right? And so the strategy that we're using is we're attaching them to monoclonal antibodies. And so we have 3 programs in the muscular dystrophy space. And really, I just bring that up today because so far this year, we've been able to show positive results across all 3 programs, right? So really reinforcing that we can reproducibly deliver these therapeutics to muscle cells in different disorders. So today we're focusing on FSHD, of course. And so that's this phase 1/2 study. I'm not going to go into the underlying molecular biology of FSHD, only to say that the way we get our RNA therapeutics, and we're using an siRNA, right, to target DUX4, is through this monoclonal antibody that targets a protein called the transferrin 1 receptor that's found on muscle cells, right? And so the antibody binds to that protein, gets pulled into the cell, and then releases the RNA therapeutics to really degrade and break down DUX4. And really the hope is that by doing this, we're halting the progression of FSHD. So a little bit about the trial. So this was a randomized double-blind phase 1/2 trial. It had 2 arms. I'm going to talk about the first arm today. And this arm was a 2 mg per kg dose, right? We enrolled patients between the ages of 18 and 65. And this was a 12-month-long study, dosing with an initial loading dose and then dosing every 3 months throughout the year. Because this is a first-in-human trial, we're really looking at safety and efficacy of these compounds to really ensure that we can safely give these to patients. And then some of our key exploratory objectives are, do we see functional improvement in patients with FSHD after delivering these compounds? So if we just take a real quick look at the demographics, right? I won't walk through all this other than to say they looked the same, really similar in the placebo arm and the Delbrax arm for the 2 mg per kg cohort. And if you look at the reachable workspace, we see that there is a slight difference between the Delbrax arm, but these patients already had some muscle weakness, right? They're about moderate muscle weakness and reduced mobility in the reachable workspace. So our primary outcome is safety and efficacy, right? And so to date, we're looking at both the 2 mg and the 4 mg arms in the safety. What we see is it's well tolerated and safe. We had no serious adverse events or no severe adverse events. No one discontinued from the drug. All of the AEs that we saw were mild and moderate. And the most common AEs that we saw that occurred in greater than 2 patients or more was fatigue, rash, anemia, and chills. The anemia that we've seen is thought to be due to targeting transferrin 1. And so far it's been transient. And so we see it initially, but then it goes away after a little while. So the first question we want to ask is, can we get our RNA therapeutic into muscle? And so this is what we show in this graph right here. So this is from a muscle biopsy from the leg. And we show that we're getting nanomolar concentrations of our siRNA into muscle. Question one, we're targeting muscle. What happens when we target the muscle? We get a result like this, right? There's no way we're going to interpret all this. The only thing I want you to focus on are the colors, right? So if we look at the panel on the left, we see two different colors, right? Each row or each column is a patient. And we can see the genes up top that are red. Those are genes downstream from DUX4 that are overexpressed. The genes in blue are genes that are underexpressed because of DUX4 expression, right? So then when we treat with with Delbrex and knock down or get rid of DUX4, we would hope to see the reversal of these genes. And that's what we see, right? You can see the genes that are overexpressed or red go down to blue, and vice versa. The genes that are blue go down to red or go up to red. And, you know, we have varying degrees across different patients, but this gives us a lot of information that we can continue to look at to understand the progression of FSHD and different molecular pathways involved. But that's a lot of genes to measure, right? So we really wanted to focus on what were some genes directly targeted by DUX4 that we can measure to robustly and reliably detect the activity of Delbrex. And so that's what we have in the Avidity panel. This is a focus panel that we've developed with 4 genes, right, that are directly targeted by DUX4, so we can reliably measure the effects of Delbrex. And we validated this with published, well-known gene panels, the Redux and the 41 gene panel. And we see a similar trend across all of these gene panels, right? A 53% reduction in these genes that are targeted by DUX4. So we're getting into the muscle, we're targeting DUX4. We've been doing all this with muscle biopsies, right? And we all know muscle biopsies are hard and challenging. So a great way to maybe move forward is if we could find a circulating biomarker that we can measure in the blood. And so we've actually identified one, and I won't tell you what it is today. There's still lots of work going on, but this is the initial results, right? Showing that when we treat with Delbrex, after 3 doses, we start to see this decrease in this DUX4 regulated biomarker. And this is really what's going to drive one of the arms of our trial that I'll talk about in a minute. To kind of show a correlation, right? If we look at creatine kinase, right, which is a biomarker for muscle health, right? We see a similar decrease in the amount of creatine kinase. So when muscle cells are being damaged, you get elevated creatine kinase. The idea is if you're preventing that damage or slowing it down, you can see a reduction in creatine kinase. So, targeted muscles, we knock down DUX4. We have potential biomarker we're looking at. What does this mean functionally for patients? So really quick, Lucine talked about reachable workspace. I'm not going to describe it other than to say, you know, the way I think about this is it's a measure of like daily function, like putting on clothes, brushing your hair, brushing your teeth, right? And what we want to see is there improvement. And so when we look at the results in different quadrants, so different ways you can move your arm, right? If we look at placebo, we see a declining over the year, right? And how patients with FSHD are able to function in this assay. And then patients that were treated with Delbrex at 2 mg per kg over the course of the year showed a trend in improvement on this functional outcome. So it's an exploratory endpoint, right? So we actually will have to run another arm of the study to validate and really determine the clinical significance of this. But it gives us some indication that maybe we are having clinical efficacy with this. So this is the study design. Today I talked about cohort A, which is 2 mg per kg arm. We've fully enrolled 4 mg per kg. And then on Wednesday, we announced that we're opening up part C. And so this is a new arm of the study that's specifically designed to validate the biomarker that we've identified in our first initial cohorts. And then in the first half of next year, we're planning to open up a fourth arm, which will be looking more at functional outcomes. So if you'd like to get involved, if you would like more information, please visit our website. Please go to clintrials.gov to find different sites. And obviously, please keep engaging with the FSHD Society. They're great partners. We work really closely with them. And these trials we could not do without information and collaboration with patients, with FSHD Society, and others.